A lot of us have dreamed and thought about what it would be like if we had an identical twin. I have already personally thought about what I would do if I had an identical twin. Maybe talk her into doing some of my chores, or maybe even some of my homework without any of my parents noticing. But the chances of giving birth to identical twins in the first place is 1 in 285. And the chances of you finding your long-lost twin is 1 in a million. And unless science has created some new cloning mechanism, you creating your own twin is impossible. Despite the very low chances of you giving birth to identical twins, especially if you were a guy, the chances of your body creating twin cells or identical cells is 100%. I'm Irene Dela Cruz, and this is my discussion on cell division in eukaryotes and prokaryotes. So before I go on to the very interesting topics of mitosis and binary fission, I will be explaining to you what chromosomes are. Chromosomes are going to be mentioned a whole lot in this video, so I'm going to get started on it now. Essentially, chromosomes are made up of one of the linear DNA molecules in the nucleus. And most eukaryotes have 2 copies of each chromosome, meaning that they are diploids, or 2N. And eukaryotes usually have 23 pairs of different chromosomes, meaning that there are 46 individual chromosomes in all. Remember that they are diploids, so then they are just copies. Matching chromosomes are called homologous. However, some other eukaryotes are only supposed to have 1 copy of each chromosome, meaning that they only have 23. Since there's only 1 copy, these are called haploids. So first, I'll be discussing cell division in eukaryotes, which is basically called mitosis. But before mitosis actually begins, there's this stage called interphase, which is the first and longest part of the cell cycle. This is when the cell is basically doing its thing and doing its job, bringing nutrients to other parts of the body, but it is also a preparatory stage for the actual process of mitosis, because there are 3 phases under interphase. First being G1 or gap1, second being S or synthesis, and third being gap2 or G2. So in gap1, basically the cell just grows in size in order to make room for the things it will be replicating in synthesis. Because in synthesis, the cell replicates the DNA that is in the nucleus and it is still within that cell, so it needs a lot of space. And in gap3, the cell again grows in size, but also copies other organelles in the cell, like mitochondria, endoplasmic reticulum, ribosome, etc. Now, some cells go through a bonus round called G0. It's basically when the mitosis process is postponed. Some cells resume it and go back to G1, while other cells don't resume at all. Now let's start with the actual mitosis process, because we've been putting it off for quite some time now. It basically happens when the replicated DNA moves to opposite ends of the cell, and then the cell divides itself into 2, creating 2 new cells. It starts with prophase. During prophase, the nuclear membrane disintegrates so that the chromosomes can actually move. And second, the chromatin condenses to become chromosomes. This is actually a very long and complicated process, but to give you a summary, the DNA molecules wrap around the centrosomes to become nucleosomes. These nucleosomes condense even further to become chromatin fiber, and this chromatin fiber condenses more and more and more until it becomes chromosomes. While all of this is happening, centrosomes are moving to opposite poles of the cell and start making their spindle fibers or microtubules. After prophase is a process called prometaphase, which basically means before metaphase. During this stage of mitosis, the microtubules grow to make their way to the center of the cell. They also connect themselves to the kinetochore of the chromosome, which is located at the centromere of the chromosome. The centromere of the chromosome is where the sister chromatids meet. After prometaphase is metaphase. This is when the microtubules make the chromosomes align at the center of the cell, also known as the metaphase plate. The next process will not happen until all of the chromosomes are aligned. And then the sister chromatids are pulled by the microtubules to opposite ends of the cell. That process is called anaphase. After anaphase is telophase. During telophase, the chromosomes actually reach the opposite ends of the pole, and then uncondensed to become chromatin again. Nuclear membranes then form around the two groups of chromatin, so technically at the time there are two nuclei in one cell. But then, cytokinesis happens. Cytokinesis is the process in which the cytoplasm of a eukaryote is divided into two to create two new cells that are exactly the same. It is seen in animal cells through the cleavage furrow, while in plant cells, they undergo cell plate formation in order to separate the two new cells. So we all know that in factories, there are a certain group of people who are assigned to check the quality of the product at different stages of the assembly line to make sure that their end product is at the utmost quality. Well, the cell sort of has a mechanism like this. It's basically to make sure that the cell isn't doing anything wrong or sketchy in their process of mitosis. So there are three checkpoints of the cell. First being the cell growth checkpoint, second being the DNA synthesis checkpoint, and the third being the mitosis checkpoint. In the cell growth checkpoint, the cells check if they are big enough to move on to the S stage. If not, the cell goes into G0 until it is ready to resume G1. In the DNA synthesis checkpoint, the cell checks itself to make sure that the DNA is copied correctly and exactly. If so, the cell moves on to mitosis. Lastly, the mitosis checkpoint checks if the chromosomes have aligned at the metaphase plate. Otherwise, anaphase cannot happen. The checkpoints are controlled by proteins called cyclins, an enzyme called cyclin-dependent kinases, or CDKs, meaning that they are only active when they are bound to cyclin molecules. Now, this is a very technical process, so I'll try to explain it as quickly as possible. The G1S cyclin binds to CDK2 that targets proteins to G1S transition, meaning the cell commits to DNA replication. Then the S cyclin binds to CDK2 that targets proteins for initiation of DNA replication and progression through S. A lot of this happens throughout the mitosis process, and there are many enzymes, there are many proteins that are involved in this, but the process happens on and on and on until the cycle is over, then it starts all over again. The cyclins are named because of their different concentrations throughout the mitosis cycle. The concentrations change. So basically, if a cyclin doesn't degrade or change their concentration, then they can't move on to the next stage because it is not compatible with the enzyme. So even if the chances of a cell making an error in mitosis is very low, it can still happen, and that's why these checkpoints exist. Some cells even slip past the checkpoints despite having an error, and they go through mitosis despite having wrong DNA. So cells with wrong DNA continue to replicate, and then they become a tumor. What causes cells to slip past these checkpoints are usually things like carcinogens, certain chemicals, or sometimes they're even hereditary, making them unavoidable. But the whole mitosis process is just how eukaryotes divide. How prokaryotes divide is a different story. Lucky for us, binary fission is much simpler than mitosis, and in fact, it takes much less time. For example, E. coli bacteria can take only 20 minutes to divide, while a eukaryote can take up to days. During binary fission, the replication takes most of the space of the cell, as it doesn't have a nucleus. Most prokaryotes have a single circular DNA, known as the prokaryotic chromosome. Replication begins in the middle of the cell, and then the two copies spread outwards. The two replicated chromosomes migrate to the two poles. But the mechanism on how this actually happens is still unknown to this day. After replication is complete, the plasma membrane grows inwards that will eventually separate the chromosomes into two cells. The processes of mitosis and binary fission happen every single day. In fact, your body has around 37 trillion cells or even more, and all of them go through mitosis. On average, our skin loses 5 million cells a day, and we use mitosis to replace those cells. When we learn about mitosis and binary fission, we see the very small mechanisms that keep us alive. Through the study of mitosis and binary fission, we see the true complexity of life. It isn't just taking a breath and going to sleep, it isn't just eating. We can see how much our body actually cares for us, how each cell is doing its best to make sure that we are kept alive. And that's what I think is the very beauty of biology. The fact that we didn't know the mechanism as to how chromosomes line into the metaphase plate until 2012 tells us that there's still so much we can do. There's still so much we can learn about all of the processes of life. I'm Irene De La Cruz, and this was my discussion on mitosis and binary fission. Thank you for watching.